right now. And sorry, they're recording. Okay, and it's really about sort of how we've been developing new analytical approaches to ask questions about dissolved organic matter composition and cycling in the ocean, and hopefully I can convince you that we have made some progress in that arena. So many of you have some version of this figure in all of your different classes. This is just a figure of the global carbon cycle. This is from the IPCC report in 2001. And, it has some of my favorite reservoirs of carbon. So you can't see the numbers, but in yellow are the fluxes in gigatons of carbon per year. And then the reservoirs are storage reservoirs. And I want to just draw your attention to a couple of those reservoirs. So we have the terrestrial biosphere, atmospheric carbon dioxide, and then my favorite, the dissolved organic carbon, or the carbon in dissolved organic matter reservoir in the ocean. And the thing that I try to convey here is that these reservoir sizes are all pretty similar in terms of, the order of magnitude, but also just very similar. And these are reservoirs that could exchange on relatively short timescales, such that in theory, the dissolved organic carbon reservoir or large changes in the size of this reservoir could have an impact on atmospheric CO2. However, that reservoir of organic carbon is sort of buffered by this much larger reservoir of inorganic carbon. And so from the perspective of influences on atmospheric CO2, I think we would really need to see pretty large changes in dissolved organic carbon. However, it is produced during primary production in the upper ocean and has the potential to store carbon on relatively long timescales, which I'll get into in more detail. And it also has it also plays an important role in structuring upper ocean ecosystems, which will also be discussed as part of my talk. When we look at the concentration gradient for dissolved organic carbon in the ocean, and this is just sort of a schematic compilation of data from off the California current here. You have DOC concentration up here in the x-axis and depth on the y-axis, just only going down to about 500 meters. And all I want you to take away from this figure is that there's high concentrations at the surface, and then it falls down to some fairly stable concentration at depth. And as you'll see in a little bit, the deep Pacific Ocean has the lowest concentration. If you look at these profiles from all around the world, really, what you find is that there's a fraction that has been functionally called semi-labile, which essentially is referring to a reservoir that's cycling. It's basically the accumulating DOC in the upper ocean that's cycling maybe on decadal to centennial timescales. And that's an important reservoir because it does store carbon on those timescales, and that's a reservoir that's available for subduction into mode waters, into intermediate waters, where even if it does turn over and turn into CO2, it's stored on some relevant timescales. And you can see maybe some of that in this blue profile, where you see higher concentrations at depth, which would suggest potentially subduction of this reservoir. The labile component, of course, which is very important in terms of structuring ecosystem interactions and nutrient cycling, is often, we can see that at the compound level, as you'll see, but it probably doesn't accumulate on long enough timescales, although seasonally we can see changes that are consistent with the accumulation of some short-lived components in the upper ocean. And then the most relevant for this part of the talk is that as you get deeper with depth, you start to see this sort of constant concentration, usually below about 43 micromolar carbon throughout the ocean basins. And we refer to this as sort of functionally recalcitrant or refractory. And based on concentration profiles, and as you'll see, based on radiocarbon, we've assumed that this component is sort of evenly distributed. It's present at a constant concentration in the ocean and up and down in the ocean, and therefore present in surface waters and a significant component of the surface water reservoir as well. And so this idea of a constant background is something that I'll keep coming back to. So this is important, as you'll see, because it is a potential reservoir of carbon storage on millennial timescales, and it might have turnover times on the, that are sort of similar to what we might think about for surface sediments in the ocean. But it's in the water column, right? And so if you were to take a whole bunch of DOC profiles, as Dennis Hansel did in this figure, you could come up with a oceanic distribution that looks like this, and it confirms sort of the discussion that we just had, which is that high concentrations dominate the surface parts of the ocean basin. And you can see that, if you follow some of the density lines, you can see that subduction of semi-labile carbon into the intermediate waters. And then the oldest the oldest water mosses, Pacific deep water, have the lowest concentrations, somewhere between 36 and 40 micromolar carbon. So the question is, what constitutes these different reservoirs of labile, semi-labile, and refractory, and other sort of structural components or handles that might allow us to understand a little bit more about their roles in the ocean, their sources, and in the case of the refractory component, what controls its residence time in the ocean. So in terms of residence time, the most sort of specific insight, I guess the most valuable insight, really has been provided by radiocarbon measurements of DOC. And these measurements were first made by Pete Williams here at Scripps in the late, I guess the early 80s, and have been continued by people like Jim Bauer and Ellen Druffel. And this figure is just a compilation of radiocarbon measurements for inorganic carbon, so dissolved inorganic carbon, and dissolved organic carbon in the ocean. So just to take a step back, what you're looking at on the x-axis is the radiocarbon. We'll think of this as a radiocarbon content. We can talk specifically about what that term actually is. But as many of you know, C14 is the radioactive isotope of carbon. And what we're looking at here is the natural abundance of that C14 isotope in organic matter. It is influenced by the release of C14 into the atmosphere in the 50s and 60s during above ground weapons testing, but it's not like a tracer that you might be thinking about when you do bacterial production measurements or something like this. So this is the natural content, or the content of C14 in DOC in the ocean today. And a couple of things are noteworthy. The first, so when we think about dissolved inorganic carbon, we see that it's enriched at the surface because it's exchanging with the atmospheric carbon dioxide C14 reservoir. And then as the waters become more and more isolated with exchange from the atmosphere, the radiocarbon begins to decay with a half-life on the order of about 5,700 years, such that in the deep Pacific, which is where these profiles are from, you see the oldest waters, right? So here we're looking at radiocarbon values of about -250 per mil, which translates to about 1,800 to 2,500 years, depending on what we think of as the reservoir age. At every depth in the ocean, dissolved organic carbon is more depleted, i.e. lower in its radiocarbon content or older than DIC. And you would expect that if primary production was the major source of DOC in the surface ocean, that dissolved organic carbon would have a signature similar to DIC in the surface ocean. In this case, because of the way we make the radiocarbon measurement, we've accounted for any fractionation associated with the uptake of different isotopes of carbon into the system. So we can compare these directly, and in fact, we would expect that to be a similar value if the bulk of the DOC was being produced primarily by photosynthesis and turning over on short timescales in the upper ocean. At every depth, of course, it is more depleted than inorganic carbon, and this has been the enigma, at least for me, throughout my entire career, starting from when I was a graduate student in Dan Rapetta's lab at Woodsville Oceanographic Institution. Now, why is the radiocarbon distribution this way? So what has been typically done is that we've explained this radiocarbon profile as simply a mixture of two sort of reservoirs of carbon, that refractory reservoir that I was talking about. So it's denoted here as delta 14 CR, And we assume that it's present at a constant concentration, but also a constant radiocarbon signature with depth, and that it has or can be assumed to have the radiocarbon content of the deepest reservoir. And then that is mixed with carbon that's produced recently from the surface ocean, so just a 2 end member model, and then the F's here just are the fraction of refractory or the fraction of labile based on concentration profiles, and that if you mix those together, you can basically reproduce the value that's actually measured both at the surface, and then as you decrease the contribution from the labile component with depth, that's why what brings you to these more depleted values at depth. And then finally, you get to a point where there's no more labile carbon, and it's essentially reflecting the radiocarbon content of refractory organic matter. However, this is not like DIC where you're primarily talking about inorganic carbon, CO2 carbonate, bicarbonate. We're talking about essentially thousands to 100,000s of molecules that sort of make up this reservoir of DOC. And so the question then has been, what is, it has to be a bunch of different ages, right? So we must be looking at a continuum of ages or continuum of radiocarbon signatures in the ocean. And so that is sort of represented here schematically as sort of very labile carbon, all the way to something we might call radiocarbon dead or C14 free. So, has a residence time greater than 50,000 years in the ocean. And in fact, if you were to expect something that had a constant radiocarbon signature with depth, then the most likely candidate would be DOC that's essentially free of radiocarbon, because it's basically made its way around the oceans, many times, so many times that it's completely mixed in the water column. And so some of my work has really focused on trying to get at the distribution of radiocarbon in the ocean, and specifically asking the question whether there is this constant background of C14, and if that's the case, does it have a radiocarbon dead component? And this has been a challenging question to pursue, but I have been very committed to it for a few decades now. So the talk is sort of divided into these two parts: the old stuff. So what is this refractory carbon? Can we say something about its structure, but also what can we say about the radiocarbon distribution in refractory organic carbon? And in the second part, I will briefly discuss some of the work that I've been doing, kind of pursuing the new stuff, using different techniques to catalog sort of the metabolites that are in the DOM reservoir to try to learn something about the interaction of different ecosystem components in the upper ocean. So one of the things that needs to be established first is that for any of the work that I'm going to be discussing here, the structural characterization work or the radiocarbon work, I typically start off by extracting DOC out of seawater. And that's because the salt is a problem for many of the analytical techniques that I use. And also, if we're looking at the complexity of the reservoir, sometimes it helps to actually simplify it a little bit to begin with. So in our typical method, you acidify seawater to pH 2, you extract it onto a resin, in this case, a resin that we call PPL. It's very broadly used in the community. And then you elute the compounds that stick on the cartridge with methanol, and that allows you to separate it from the salts in seawater. The disadvantages of this method are, of course, the acidification itself, the fact that it's a chemically specific extraction method. So you're focusing your efforts on hydrophobic or semi-labile compounds, and you're going to miss the very polar compounds. In shallow waters, the recovery is as good as 40%. So 60% of the DOC is blind to some of the methods that I'm discussing downstream in this talk. But at depth, we can get higher extraction efficiencies. We get about 60%, but others have been able to get even greater extraction efficiencies in the deep ocean. It's compositionally different as well from the bulk, which is to be expected given that it is a specific sort of chemically specific extraction method. The advantage in the context of the radiocarbon work is that despite the fact that it's only 40% efficient at the surface in the surface ocean, and it's chemically selective, the radiocarbon content of this extracted component mirrors the bulk component throughout the water column. So at the surface, we have values between -280 and about 300 per mil, similar to the bulk reservoir. And at depth, we have values around -550, which is what we expect for the deep Pacific. So that's been interesting, and that has allowed us to, well, that has led to us assuming that both the refractory component and the young component must be represented in some way in this fraction that we extract from seawater. And this is sort of what I just wanted to show this for the students. This is what it really looks like when you're doing this at sea. This is my student Ralph Torres. We were recently out for 4.5 weeks in the California current ecosystem. The little columns that you're looking at here are the ones that we use for metabolites, which is why we have so many of them. And then sort of we use these bigger columns for the radiocarbon work because we need more carbon. So another thing that we've been able to develop over the last couple of years is a method to then take that extracted DOC and further concentrate the older material. So we're really going after this refractory component in this particular case. And so the idea is that you take that extracted material that's made up of all these different end members, you do an acid digestion and you re-extract it again on PPL resin. You lose different amounts of carbon depending on where you are. In the surface, we lose about 40% of that PPL component, and at depth, we lose very little, maybe 5 to 10% total. And that suggests that at the surface, you have more acid labile material like sugars and amino acids. And in fact, we've looked at the components that are not retained the second time, and they are rich in those types of components. So the idea is that once we do that, we're really concentrating the older material, and then we can look at that by itself. And we have evidence to show that this is in fact isolating the older material. So again, this is radiocarbon in the surface with depth now down to 4,000 meters. This is a profile at Station M in the Eastern North Pacific. And in blue are the radiocarbon values for the first bulk extraction of PPL. And these are the values that I said mimic what we expect for the bulk DOC that's not extracted. And then in red are the values for the component that we concentrate using this acid digestion approach. And you can see that at the surface, you get significant removal of that kind of labile component, as you might expect, and you see a decrease in the radiocarbon signature. And at depth, you see a decrease as well, but it's not very prominent. And in fact, we really don't see values that exceed -550 in the deep ocean. So we are, if we were to take these data together, we would say that for the hydrolyzed or the acid digested component, we do see it isolating a more refractory component, as suggested by the decrease at depth. But in general, we still see a gradient for this refractory component, i.e. it's not fully mixed up and down the water column, and we don't see evidence of something that's radiocarbon dead. So the most significant decrease is observed in the surface ocean, but there's still this vertical gradient in radiocarbon, and we don't see any values that exceed -550 per mil. So there's no evidence of this dead component. And you might say, well, is this sort of a common observation to have this gradient? Are there other studies that have shown the prevalence of a component that's more dead than this? And in fact, if you, sorry, I have to use my animation. So yes, there's no dead component, and you clearly have a mixture that's removing the younger component with the hydrolysis. And so if you were to compile the few studies out there that exist that have tried to isolate refractory DOC using different methods, we consistently see this gradient with depth. The upper ocean values are very similar, regardless of when this was conducted or where it was conducted. And the gradient exists with very little evidence for anything, in this case, maybe less than -600 per mil. And so this appears to be a consistent observation, which is very much at odds with the paradigm that we typically use to explain the radiocarbon distribution in GOM. And it raises some interesting questions, many of which remain unanswered. So one of the other aspects of this was to say, all right, well, we have this gradient with depth. Does it look the same? Does it look like a background component? Is it structurally consistent? And that's been challenging to try to answer that question, because the techniques that we have available to us have a hard time dealing with this particular fraction. And so that's what I'm going to talk about in a little bit. So I just wanted to point out that some of the kind of long-standing assumptions has been that the 14C depleted component, it has a sort of a constant concentration with depth, and we really don't see evidence for that, and it's consistent with others, the work of others. And we don't see evidence of a component that's radiocarbon dead, something that's also been invoked to explain sort of this idea of a constant background. So one thing we wanted to investigate, as I said, was to determine whether this background component that has this gradient has some similarity in its composition. So for example, if the surface water composition is significantly different from that at depth, then that would suggest that maybe we really do have a significant mixture of young and old material at the surface that's still giving us this appearance of a radiocarbon gradient. And to some extent, we would expect the composition to be broadly similar because we're using this chemically specific extraction method. So as I said, this is a complicated thing, and we've approached this in two different ways. One is to use something called RPO or ramped pyrolysis oxidation, which I'm going to talk about briefly. And that's been broadly used to study sediments and soils. And really the reason we went after this technique was that it's been used in these other studies to explore the radiocarbon distribution in organic matter. And I'm not going to present those results today, but I'm happy to talk about what we found and why I'm not discussing them here today. So this is, I'm going to talk through this very quickly. I'm happy to talk more about this with those who might be interested in this. But it's a very simple idea. You have an oven that ramps from about 200 to 600 degrees. And so you see this temperature ramp, temperature increases with time. So it basically burns the compound in a stream of helium and oxygen. So in this particular iteration, this contains oxygen so that you can oxidize all the way to CO2. And as that CO2 evolves, it is measured and quantified. So you get a measure of CO2 evolved over time. And then if you want to trap different fractions of that carbon dioxide over time for radiocarbon measurements, as I said, to get some sense of the distribution over this thermal gradient, you can do that and trap it for radiocarbon, which we had done in some cases. But this particular part of the system where you can measure the CO2 evolved over time can actually be, you can take that data and convert it using an Arrhenius form to activation energy distribution. So the idea is that you can get this thermogram, so the CO2 evolved over different temperature bins, and utilize that to get a sense of the activation energy distribution in your sample. And this is a very old technique. You might have heard about calorimetry. So the idea here is that somehow the composition of the sample is related to the thermal profile of that sample. And so here's the data that you typically get out of this instrument. So you have carbon dioxide normalized to the starting carbon amount, and then temperature. So this is what we typically refer to as a thermogram. And in this case, I'm just showing you the thermograms of what we get for that first PPL fraction, not the digested one, but this is what the thermograms look like for the original extract. And I won't get into this too much, but I'm happy to talk about it. And so you use that Arrhenius formulation and you can get a distribution of activation energy. So there's two columns here. The one is looking at that original PPL DOC extract. And here I'm talking about the more refractory component that's isolated after acid digestion. And on the x-axis, you're looking at activation energy distributions. And on the y-axis is just a quantification, the percentage, the fraction of carbon that was present in the sample at the beginning. So what you need to really, what I'm really trying to draw your attention to here is the distribution of activation energies in these two fractions with depth. And the main point I want to make here is that when you take that refractory component that's isolated after acid digestion, you see a fairly similar profile in terms of the activation energy distribution with depth, which we are interpreting to suggest that it has a fairly similar composition with depth. And it's not to say that's the identical compounds at every depth, but that the mixture of compounds in each of these fractions, regardless of the depth, together give an activation energy spectrum that's very similar. And so there's elements, structural elements that must be quite similar across these different depths. And you might say, okay, is there really, what can you really learn about chemistry this way? And we're still working on it. So one of the very recent additions that we've made is that we've started collaborating with Zamfir Liu at UT Austin's Marine Sciences Institute, where he uses the same sort of thermal fractionation approach, but then has a much more sophisticated structural characterization tool of essentially a mass spectrometer at the end of this. And so all I want you to take away from here is that at these different temperature fractions, you see compositional differences, right? But it's also important to keep in mind that thermal degradation is a real thing. And so what you're seeing here at these very high temperatures, which are just dominated by aromatic compounds, is very likely a result of the actual temperature ramp. So it's not making new carbon, but it's making new kinds of carbon because of the pyrolysis method. And I know it's a sort of a very quick introduction into this, and I can talk more about how we interpret this if you're interested. But I just want you to get a sense that indeed different types of functional groups are kind of burned off at these different temperature ramps, and that there is also this alteration of the sample likely as the pyrolysis takes place. So this is one way to get at composition, and I'm going to sort of rush through this next slide and I can talk more about it, but it is published work. We've also tried to utilize some other types of chemical degradation techniques to get more specific information on the chemistry of this refractory component in the surface waters. And what we found is that the oxidized products of lipids in particular appear to be an important component of this refractory fraction, or the functionally recalcitrant fraction. And by that, I just mean essentially lipids that are decorated by different oxygen moieties. They don't have to be identical at every depth, but the general composition appears to be quite well preserved. And I'm showing you this schematic that's sort of, it's in the paper. And the idea here is that we tried to do this with a model compound. We took carotenoids, we exposed it to light, but also reactive oxygen species would do the same thing. And you end up with, from one compound, you end up with hundreds of compounds that are structurally similar, but not identical. They have sort of backbones that might look like this, and they're decorated with hydroxyl and carboxylic acids. I don't know exactly where those hydroxyl and carboxylic acids are because we can't detect these in their native form. We actually do a reduction and then measure detect just the carbon skeletons, but we know they're oxidized based on nuclear magnetic resonance spectrometry and some other tools that we use. So essentially, to put this all together, we think that the hydrolyzed component that we are observing by RPO may have a native structure, something like this, or related to this. And there's many potential kind of isomers or related compounds that can have this sort of major functional diversity. But we do think the oxidation of these lipids is an important component of what's accumulating in the long term. And the power of doing this sort of model-based approach as well, and comparing that to what we find in the environment, is that we now have sort of a model that we can use to look at the processes that might form refractory DOM and why these might be long-lived, why they may not be, why they might be functionally recalcitrant from a biological perspective. So the conclusions for this part is that 14C measurements for the refractory component exhibit a depth gradient, which is not consistent with our hypothesis or the expectation that it's stable or it's a constant across different depths in the ocean. We don't have any direct evidence for the presence of a significant fraction of DOC that's older than -400 in the surface ocean. So again, this gets back to this depth gradient. Both observations call into question the existence of this constant background. The chemical similarity of this refractory component is evident from the thermograms, and this is consistent with our expectation that it is sort of a background fraction. And by characterizing the operationally defined component, we suggest that oxidized lipids are important contributors to long-lived DOM. And there are implications with this for the this value of -400 at the surface and the fact that there's no dead radiocarbon. And I can speculate on that, but again, that's ongoing work, and we don't really have any conclusive evidence for how this really, we know that it alters our idea of how DOC is cycled in the ocean, and it has to impact the residence time, but exactly how to interpret that is work that we're ongoing, but I'm happy to speculate and discuss with you more. Okay, so I'm going to switch gears here a little bit, and I know I have about 15 minutes, I think, before I should end, and I'm going to talk a little bit about the new stuff, how we sort of explore what analytical tools we're using to really uncover the chemical diversity of metabolites in the surface ocean. So I want you not to have expectations that I'm going to say, oh, these particular compounds drive these particular individual interactions between these organisms. But what I'm going to try to convince you is that the tools that we're developing allow us to really expand the chemical space in terms of what we understand about the different metabolites that are in different environments. And that will hopefully lead to a better understanding of how these chemicals mediate these interactions in the upper ocean. So in this case, we start off with that same PPL extract, but we separate that extract by liquid chromatography, and then we use tandem mass spectrometry. So for those of you who care, this is the analytical approach. And essentially what you get at the end of the day is a series of ions that are coming out that are separated by time and polarity, or whatever you want to separate it by, depending on the column that you use. And then at different times, you can isolate these ions and bombard them and fragment them and get at the structure of those compounds based on the way that they fall apart. So this is very important. We rely on the fragmentation of these features or these molecules to, if we don't have definitive identification, we use that fragmentation to essentially use machine learning to predict the structure. And that's hopefully what you can get, what I can convey. The disadvantages of these techniques are that you're using one technique it's very biased, right? It's only as good as the things that it can see. And it's very hard to determine what you can't see, right? So you can only talk about what's detected. Even quantifying what's detected relative to the total is difficult with many of these approaches. And then the chromatography with liquid chromatography is not very good. So you're not really separating out compounds you don't get excellent separation of compounds, and that can complicate the data set because each compound influences the other. But the advantage is that it's really very high throughput, so we can do kind of metabolite maps that match the 16S, 18S maps that we can also generate. It's widely available to the community, so it's a very accessible technique. And the informatic methods, because it's broadly used in the health sciences and the pharmaceutical sciences, the informatic methods are highly developed, and that's really, we've been taking advantage through collaborations with folks on the main campus at UCSD who are in the pharmaceutical sciences. So what types of molecules might we be interested in identifying? So, you can imagine that organic nutrients are particularly important, maybe in oligotrophic systems. We know that vitamins, for example, are only produced by certain organisms, but required by everyone. Signaling molecules might tell us something about the response of the community to a particular environmental condition. And being able to identify who's producing the signaling molecules would be really fantastic. And then things like toxins. So domoic acid is a toxin that we're particularly interested in on the West Coast. It's produced by the diatom Pseudonychia, and we've done some work with that. And you'll see this example over and over again because it's an easy one to detect with this method. And there's different ways to approach these questions, right, of interactions and metabolites that drive these interactions. And this is a nice sort of schematic by Brendan Durham that was published earlier this year. And we're on this end. We're really going into the environment, into the field, trying to isolate really the entire metabolite reservoir that's accumulating and doing kind of an untargeted approach. So really it's really, people would like to call it a fishing expedition, and in fact it is. It's a hypothesis generating expedition. So we've certainly come under criticism for kind of taking this approach of going into the field, but that's really where our interests are, and that's kind of where the challenge is for us. And then you can do sort of more controlled studies in the lab where you have a particular organism and maybe a particular bacterial counterpart, and really excellent insight has been provided in those kinds of studies. And you can sort of try to do this computationally with microbe metabolite networks. And I'm going to talk about our work from field samples here. And so I'll just get into it right away. This is something that others have used the term interactome. But what you're essentially looking at in a very small space is correlations between 16S and 18S sequences and metabolites as detected by the methods that I just discussed previously in different types of environments. So we didn't necessarily expect them to fall out in this sort of in an environmental specific way, but what you see in the red is an environment where it's highly productive. It's recently upwelled water in the California current. It's rich in diatoms, nutrients, and iron, of course, are there, and that's why you have a big diatom bloom, and you can see the species composition is consistent with that. And then you also see particular metabolites that are dominant or correlating strongly with the organisms in that environment. If you go to an aging ion-limited system, so it's that same water moss, but it's aging, so the nutrients are slowly getting drawn down. It's extremely ion-limited. There you see different kinds of organisms and interactions with metabolites that were unique. And then you can, go out into further offshore where you're both ion and macronutrient limited, or you're in a fully chronic macronutrient limited system. And again, you see these different communities falling out and correlating with different metabolites. And then there are there is a group of metabolites and organisms that are correlated across many of the same that show up in multiple regions. So you can zoom in on these, right? And you can see that it's really important to be able to understand what the structure of that metabolite is. So here's an example of what we might expect, what we would have expected, and it shows up nicely in the data. So it gives us some confidence in our correlation approach here. So we have a Pseudonychia 18S, Pseudonychia 18S sequences are strongly correlated with a chemical that we detect that is identified as domoic acid, which is a toxin that Pseudonychia produces. We also see some domoic acid degradation products that are linked to the main node of the metabolite. And you also see that another diatom is present in this network, and you might ask, is there some relationship here? In the aging ion-limited community, we were surprised to find a network that was dominated by a fungal Mycosphaerella. And what we found was that it was actually, it was in a network that was correlated to a metabolite that did have an analog hit and later confirmed as diacetoxyscarpanol, which is a mycotoxin so produced by fungi. And it's in this particular network, and you can see there's an unknown metabolite also correlated. In a more offshore environment, we see a compound that had an analog hit, not a library hit, but the analog hit was for indole acetic acid, and by looking at the mass spectrum, we were able to annotate it as indole butyric acid, which is also another known signaling molecule, and it appears in this network with a bunch of different groups, including a protist. But what we find is that when we get, much of these networks are very hard to characterize. So 16S data allow us to identify the organisms that might be present, but almost all of the metabolites in this network that's highly correlated don't have library hits or analog hits. So we don't know what they are. And in fact, if we want to ask questions about these ecologically meaningful interactions, we need to know something about those metabolites. And that's really been one of the big pushes in my lab recently. So here is essentially a figure, and I apologize for it looks like hairballs, but it's a figure that visually represents the chemical diversity in this sample that I just kind of took apart as individual correlations. But here now we're just looking at only the chemistry, only the metabolites. If the compounds are chemically related, they sort of cluster together in these subnetworks. The different colors in this case identify different locations and different days. And so you can color it any way you want. And this just shows you sort of the diversity of chemicals in the environment and kind of the fact that they are specific in terms of spatial and temporal variability. So in this data set, we have about 4,000 nodes. And the problem with these kinds of data sets is that, you might have a subnetwork, so it would be one of these that's blown up, where you have one compound that is a library ID, but really only about in this case it's Spink, so the library IDs are these two compounds in this subnetwork. It's only 4.7% of the total data set that has a confirmed and confident library identification. Based on what's in the data set, what's in the libraries, this approach can also say, oh, we've got a molecule in there that has a really strong relation, chemically very closely related to a library hit. And that's shown here as an analog hit. And that expands the data sets to about 12.3%. But even in this network, you can see that there are related molecules that don't have library or analog hits. And as I said, much of the data set remains sort of dark in this context. And so we've been trying to use informatic methods to sort of advance the characterization of these unknown networks. And in some of them, you can see that many of these subnetworks don't even have a single library or analog hit. So they might be really chemically very interesting, but we don't have a way to know anything about that chemistry just by relying on library and analog IDs. So we've been using informatic methods that others have developed to really address this question and try to kind of increase the chemical space that can be annotated. And I'll just show you one example here. So we can start with something that's known. As I said, we have two library hits in this subnetwork and an analog hit. Here's the known structure. And then you have some compounds for which you have the mosses, but you don't know really anything about it. And so you can use some of our informatic techniques, and just focus on the domoic acid network for now. And you can learn a few things. So for example, you can see that all these nodes that are colored in green are related somehow to amino acids and derivatives. It also has pyrrolidine, so that's this particular structure that's shown for domoic acid. It's also kind of broadly defined as an organic heterocyclic compound because you see this heterocycle from the pyrrolidine, and these are sort of independent level identifications. We also have methods that give us the molecular formulas for these nodes as well. And so you could get some confidence to propagate your identifications based on the relationship that we see to known components in the data set. However, here's a network that appeared only in two particular samples that we were interested in, and we have no library hits or no analog identifications. And so we have to kind of rely completely on in silico annotations. And we find that the network has two particular components, this organic heterocyclic component and these benzenoids that are very unique, that seem to dominate all the nodes that are in the network. And if we look at some of the structures that are predicted for this, the substructures that show up quite consistently are ones that are quite unusual. In this case, the benzene ring is not unusual, but this particular ring structure is relatively uncommon, at least from what I have seen in the literature. But that appears to be a structure that's consistently predicted for this whole network. So we can go in and say, what are the compounds that we know of in nature that contain these particular moieties, and we can try to put that in the library and see if we can then generate library or analog hits to this particular network. And so there's a lot still to be done to validate this, but it gives us a starting point for a network that before was completely dark in terms of its structure. And so using this kind of approach, we can essentially almost populate every subnetwork that's in this data set. And so we can get up to about 56% where we know something about chemical information. And if we take the relationships within a network and walk it out to the rest of the network, we can increase it as much as 75%. But I would say the 56% is what we can do with some confidence. And I can talk more about how we establish confidence in a little bit. So the conclusions for this section is that we can identify now metabolites that play important roles in upper ocean ecosystem functions in this untargeted manner. As you can, of course, unique compounds mediate interactions, but then we find that there are ones that are important that are not well represented in our mass spectral databases. So we need new annotation processes in addition to very controlled studies in the lab as well. And so combining these approaches, the field culture and structure prediction tools, we can try to expand our understanding of the chemical diversity that might mediate these interactions in different environments. And if there are interactions that show up over and over again in the same environment, right? And then, as I said, we can use these approaches to kind of populate 56% or so of these networks before we are only getting about 12% identification. And so I'm going to stop there, and I'm going to thank a couple of people in my lab, so several graduate students, and Daniel Petrus was a postdoc with me, who's now in Germany. And then all my sequencing data that I showed here was generated by Andy Allen's lab at the JCVI. And then all our mass spectrometry work we do in collaboration with Peter Dorstein on the main campus and Sebastian Bocker at the University of Jena. So I'm going to stop there and thank you for your attention.
Thank you. That was great. If we have any questions from any students or postdocs, that would be awesome. I have a question about the RPO method, potentially. Is there a reason that you're only going up to 600 with the RPO rather than higher?
I think it can go up to 800 degrees the way it's constructed, but we really don't see, we don't, at least with DOM, we really don't see a lot. And black carbon comes out around 650. So you don't get things that are much more thermally stable than that. So it's about where even in particulate samples, you really don't see things coming out, much carbon coming out past 650 or so. And sediments is the case too.
Great, thank you. All right, if there's no other questions, we can open up to faculty, and I think Bethany has a question.
Hey, Lihini, what a treat to see you.
Thanks.
And your background's killing me because summer's starting to end here. It's
I know.
I'm like, oh man, look at that beach. That was a great talk, and obviously the end, the sort of second portion is just stuff that I just love to think about. And one of my really simple questions is, and given that you're collaborating with the Allen Lab, do you see kind of different types of association when you start getting into either proteomic or gene expression data in those networks? And have you done that? So rather than looking at sort of all the taxa, kind of reducing it for whatever, you had that beautiful diagram of, your different ecosystems and what might be going on in the different nutrient status. And obviously we see sort of canonical iron limited genes, for example. And I'm just wondering if that tells you a different story or if you've looked.
We haven't looked, and it's a great question. I think that's it. I would like to say it's an easy next step, but it turns out like, trying to get transcriptomic data on those large data sets, it's not as easy as it probably should be in these days, in this day and age. But I think that's, that's definitely where sort of Andy would like to see some of those things going, because I mean, the big issue here, right, is that, what people want to know is how can you validate, right, those correlations that you're seeing. And it's not really, you can pick your favorite system and try to recreate it in the lab or something. But if you had an additional layer, such as with gene expression, that would give us some confidence, those correlations have some meaning, and maybe even tell us something about, maybe even, what we would really like to see maybe is go and look for biosynthetic pathways, right, that we predict would be there for those compounds, and see that those are correlated as well with the, but yeah, it's a great question, and I'm happy to, have people do those kinds of things and just send us the mass spec samples to analyze.
Yeah. So it really sort of opens the box for sort of looking at, I love the fact that you can sort of see signaling as well as, as these metabolic products. So thank you.
Thanks, Stephanie. Dave.
Yeah, I have one. Hey, a follow up on Sam's question about the RPO method, where you saw all the aromatics coming out at 600 degrees. And that, one, is that consistent in all the water samples that you do? Is that
it's so cool. Like these are, I just get like caught up in these little side stories, and I do nothing really that's productive, and my students want to kill me. But so what's really interesting about DOM is that there's the highest temperature peak that we see is always in the surface. It's gone by the deeper waters. And if we do that scrubbing to isolate the refractory component, it's not there anymore either. So it's clearly, and if you take all the RPO samples that we have from the surface ocean, it actually scales with bulk radiocarbon and DOC concentration. So essentially there's this very high temperature peak that shows up, and we think it's being made from something because we can remove it, right, with this, and so I don't, we don't know what it's being made from, but it's definitely being made from, I think, and something that's burning early. And the other thing to keep in mind, which I have to keep reminding myself, is that what's most representative of what's in the sample, like in the native sample, is what you see coming off at those early temperature peaks, right? Because that's it hasn't like had time to get transformed in the RPO. But then the later temperature fractions are going to reflect kind of potential thermal alteration. And there hasn't been enough conversation about that, sorry, yeah.
Are there some things that we know that would come out at 600 degrees that would remain intact? And so, and then make the conclusion that those things aren't in the ocean?
Yeah, so that's a great question. So one thing could be some sort of lignin-derived component. Others have suggested black carbon is really important, but we expect black carbon to kind of accumulate in the system. So it would be interesting if that's black carbon. It's suggesting that it doesn't survive with subduction to depth. So which is, I think, really interesting. Yeah, and it could certainly be there for sure. Yeah.
Great. Well, thanks. Good to see you again.
Yeah, it's great to see you. Other questions?
Yeah, I have a question.
Hi.
Hi, a great talk. Thank you very much. I was trying to think about linking the two parts of your talk together. The metabolites, are all of those considered labile, or do they also break into these categories of non-labile and or labile and refractory?
The sad thing is that most of the stuff that we can extract is not labile, or it wouldn't fall into the metabolite category. So it's actually people are surprised that the PPL method even pulls out some of these molecules that show relationships to communities. And the hard thing is we don't have a good way to quantify how much of that is sort of labile. I think where, what we do see are certain molecules that show up all the time and all across our data set. So those are correlated with certain types of organisms. But there's definitely a large fraction that's refractory in that, and that's kind of consistent with the extraction method. So,
okay.
Yeah.
Yeah, I was thinking about then if your map of your evolution of kind of bloom to nutrient limited, if that was also like a labile to recalcitrant transition, but it sounds like most of it is recalcitrant.
Yeah, and we see that with depth, but what's really interesting is that the DOC concentration, well, in our systems, right, like in oligotrophic systems, the DOC concentrations are actually the highest, unless you're in the middle of a bloom and like zooplankton are just like chomping. You don't really see very high DOC concentrations in our coastal systems because you're constantly diluting with this deeper water. So it takes time to see that accumulation signal in the DOC. And so as a result, the radiocarbon content of bulk DOC is highest in the oligotrophic systems. And so there is this kind of the functional recalcitrance of DOC changes, as you might expect, right? Depending on what the conditions of the environment are. So you actually kind of get more labile carbon in a way offshore, but it's not labile to the communities that are there or not to the same extent, maybe.
That's cool. Thank you very much. That's great.
Okay, well, if there's not any more questions, well, thank you again for such a great talk. It was awesome.
Thanks for the invitation.
Yeah, of course.
Good to see.
Yeah. And I hope everyone joins us again for our next talk, which I think is on the 12th. So hopefully see everyone then.
Thank you.
Thank you.
Great.
Thanks, Bethany.
Thanks,
Bye.
Lihini.